So we've got our 2-photon microscope. We've got our fluorescent calcium indicators that work with it. We can put these dyes in a sharp electrode, we can stick them into a cell, and we can image what the cell is doing. But it's tempting to say, why go to all that problem? Do you know how much a 2-photon laser costs? I don't these days, but right, a sensible number might be $100,000. That's a lot of money to spend when you've already got the sharp electrode inside the cell, which is providing you with a better accuracy recording. The only thing you can't quite image is what's happening in the dendrites, and we're not getting perfect, ratiometric estimates of calcium indicators. So why would we really want to do that? Maybe doesn't seem like a big advantage. So Arthur Conniff came in with the AM ester, right? So we already had the AMSs. They were invented in 1981 or 82 or something, but Arthur saw the use of them. And so this looks like a sharp electrode, but it's just an injection pipette. He stuck it into the brain, he squirted in this AM ester, and then he came back in like half an hour or an hour. So remember what the AM ester does. It goes everywhere. It's fluorescent. It's really doing calcium stuff, but it's doing inside cells and outside the cells. But because it's fat soluble, it can go into cells, then it gets cleaved by the esterases and it ends up being water soluble, and now it can't escape the cell. But slowly, the blood supply is washing the stuff that's outside the cell away. And so after an hour or so, you get images like this. So here you can see 1, 2, 3, 4, 5, 6, 7, 8 cells at 60 microns down. See, because he can focus down with the power of this optical sectioning power of the 2-photon microscope, where there's no out of focus light causing problems. Focusing further down and further down. This will be in layer 2, 3 by now here. You can see a little blood vessel there. Blood vessels are often coated in astrocytes that like to absorb these dyes very much. And now he can record the um, calcium transients generated by all of these cells. And when I say calcium transients, I mean these peak in the brightness of the calcium signal that we believe to be correlated with action potentials. And so now he has the ability, you can see this image there. There's going to be, probably 50 cells or so that have good calcium transients in this image. So, injecting a dye is good, and it has lots of use. But one problem, right? One problem that I mentioned is that astrocytes and neurons will take up this injected dye. So when you're looking at a cell, how do you know it's a neuron or not? If it's silent, maybe it's just because it's an astrocyte and doesn't fire action potentials, right? Or maybe it's an inactive neuron. If we had a genetically encoded calcium indicator, a GECI, which is not an acronym I like to say out loud, we could get it to be selectively expressed in neurons. And so there were lots of attempts. People tried this for a long time, and the first one that was pretty good was something called GCaMP. G for genetic, calcium, the Ca for calcium, right? And basically, we're still doing the same thing. We get EGFP, which is a fluorescent protein. We bind it to calmodulin, and we get a calcium indicator. And this is what you see. So this is where a stimulus was applied, and you can see the brightness increasing from 0 to 0.5, which basically means it got 50% brighter. That's what this delta F over F is. We'll talk about that in a second. And so, it lasted for about 500 milliseconds, so half a second, which obviously well outlasts the length of an action potential, but still, promising. Now, they kept fiddling around with it and made it better and better, and GCaMP 3, so the third version, was a big improvement, and this was the first one that saw widespread use. And these are sort of the kind of things that I would expect to see if I was doing calcium indicator work even today, right? So they're making a cell fire 1, 2, 3, 5, 10, 20 action potentials, and you can see the brightness get bigger and bigger. Obviously, so this is delta F over F. Oh, this is in percentage, so 500% brighter than baseline versus one extra potential, which is sort of 10% brighter than baseline, and you get a pretty nice linear response, which means you can kind of infer the number of extra potentials from the delta F over F, the change in fluorescence over the baseline fluorescence. And here's just a graph showing how much it's an improvement over GCaMP2. People didn't stop. So GCaMP 6 was sort of the next big improvement that lots of people used, and that was the one I've used extensively. And here you can see that you're getting a 30% increase in response to a single action potential with GCaMP 6S. You can see it comes in M and F, right, which is slow, medium, and fast. So you can see the rate at which it decays changes. Now, the fact that the slow one is always brighter made me always use the slow one because it's a rare time when these kinetics actually matter. Um, but you can see now that we get a very clear signal in response to a single action potential. So why is these so good, right? Why genetically encoded sensors so good? Well, as I said, it allows you to selectively label neurons, but not just neurons. You can selectively label different type of interneurons, or you can selectively label the glial cells if you want, or you can select the cells that express other neurons like dopamine, or noradrenaline, or serotonin, or histamine, or express any other protein. So long as they express some gene, you can probably control it. And how can you do that? Because of something called the Cre-recombinase system that I hope you've heard of in another class. But basically, the Cre enzyme gets the Cre-recombinase enzyme. Uh, you can make it so that enzyme is expressed under a promoter of something. So in 311, we learned about parvalbumin positive interneurons. So let's say you make a cell line, you make a line of animals, sorry, that expresses Cre-recombinase under the parvalbumin promoter. So only cells that express parvalbumin will express Cre-recombinase. Okay, so what? Well, what Cre-recombinase does is it gets a section of of genetic material that has LOX P sites, or special sections of genetic code on each side of it, and it flips them around, right? So what you do is you put your gene in backwards, and then if that cell also expresses Cre-recombinase, Cre-recombinase will see this backward set of DNA with these LOX P sites and flip it around, and then it will be expressed properly. So you get a Cre-recombinase expressing animal that expresses it in the cell line you want. Then you get an animal that expresses GCaMP6, FLOX GCaMP6. So in that animal by itself, there'll be no GCaMP expressed at all, or very, very little. But then you breed them together, and now all of a sudden, that wherever that Cre-recombinase was expressed, you get GCaMP expressed. Now, that's quite nice. An alternative approach is you inject a virus like an adenovirus that contains FLOX GCaMP6 into the brain. So the virus transfects all of those neurons in the area around where you injected it, but only those cells that express Cre-recombinase will correctly flip the GCaMP so that it can be expressed.